get right into it. So right now we're going to be getting started with biochemistry. As you can see on my screen, I have a blank PDF open where I'm basically going to write down whatever I need to, or use a pencil to draw anything I might need to draw. I can also look up things on my other monitor right here to copy paste into here and show you guys. On my lower screen, I have my annotated first aid, which I can't have on the screen, but you can definitely go ahead and shoot me an email if you have any questions about getting a hands on it or purchasing it, anything about my courses that I run or whatever else that you might be interested in, as well as any questions about studying tips and whatnot, which I've already posted videos about as well. So for biochemistry, the first section is molecular, where in First Aid 2021 they definitely added a couple things in that I already had mentioned in the First Aid 2020 annotations that I previously had. The conversation starts right off with chromatin structure. So they write that DNA basically exists in the condensed chromatin form to fit into the nucleus, and they give you this general information about the things that make up the chromatin structure, right? So you have your DNA, which you need to know that it makes two loops around the histone octamer. You have to remember that, and those two loops around the octamer is what creates what we call a nucleosome, and that is the beads on a string image that they're showing you on this first page over here. Those loops that are going around the octamer are held in place by a linker, right? That's the H1, right? The H1 linker is what's holding it all together. They're showing you all of that really nicely in an image, and you can see how from the top to the bottom in this picture, it's a DNA double helix that's becoming more and more condensed until you see the metaphase chromosome, right? And then they also label for you in the picture the different parts of that octamer, right? So it has 8 things to it. It has 2 each of H2A, H2B, H3, and H4, and then there's that one linker called the H1, right? So you have to remember that entire thing. The other thing you need to know from biochem about this entire chromatin structure is the fact that there's a bunch of phosphates, right? So phosphates are what give DNA its negative charge, and you always have to remember that. Lysine and arginine on histones are what give the positive charge, right? And because they have these opposite charges, they stick together and they nicely become very condensed. So there's a mnemonic I had made up where I wrote histones are large. Now if you write it like this, if you write histones are and then large like that, and you could see the L and the R's right here to remind you of lysine and arginine. As far as step 1 biochem is concerned, you have to remember that there are a bunch of step 1 topics that can never really be tested on CK because there's no time for that, right? CK will never ask you about the lysine and arginines of histones. It's just not a thing when it comes to CK. Can it be a topic? Of course, it could be a question. It's fair game, yes, but it's very, very unlikely that you'll have that as a question. So here on step 1, it's that much more likely that it will be the question, right? You can definitely have that as a question. The lysines, that big L that I've separated, are particularly important because in gene transcription is actually often regulated by covalently adding or removing methyl or acetyl groups on these lysines, which actually changes their affinity for DNA. So when this lysine no longer has an affinity for DNA because you added some kind of methyl or acetyl group, whatever the case might be, then they don't stick together, right? So we just said that the negative charge of phosphates and the positive charge of lysine and arginine is what causes them to stick together, and then the DNA is no longer accessible to enzymes for you to go ahead and do transcription and then eventually translation. So by adding acetyl or methyl groups to the L, that lysine, you can cause the histone to either like the DNA more, thereby making it tighter and then no longer available for gene transcription, so that will be methylation, right? Methylation mutes. Or you could make the histone not like the DNA, and then it's going to uncoil, right? Thereby allowing accessibility for enzymes to actually attach to the DNA and do transcription. So that's what we see later on, right? So the rest of this page talks about heterochromatin, euchromatin, DNA methylation. Then there's a separate topic of histone methylation, histone acetylation, and histone deacetylation. What we just talked about is about histones being acetylated or deacetylated or methylated. These are all 3 different case scenarios. Those are the last 3 things you're seeing right there. So let me actually put that up here too, that we're using First Aid 2021, right? So that's a pretty key thing for you to follow along. So right now we're looking at the fact that histones have this lysine and arginine, right? So lysine and arginine, key thing. Lysine more important for acetylation / methylation. So this is all about histone acetylation, and histone methylation. Really key thing to remember. You're going to get answer choices which are going to be about both histone methylation and histone acetylation and so on. And then they'll also have the answer choices of DNA methylation, DNA acetylation. That's a little different of a topic. The histone thing is a little more significant right now, actually a lot more significant. That's the one that we really care about. When we talk about something being methylated and muted or acetylated and activated, we're talking about usually histones, not the DNA itself. So as far as definitions are concerned, the rest of the things you can read on your own. But as far as histone methylation and acetylation are concerned, those are very critical things clinically and as far as your test questions are going to be about. Histone methylation causes reversible, so you can undo it, reversible transcriptional suppression, but can also cause activation depending on the location of methyl groups. Now that's pretty critical, but as far as step 1 is concerned, you really don't care about the fact that it can also activate. We just need to remember methylation mostly makes DNA mute, but we're going to just remember methylation mutes it. That's all we really care about, right? Because if we're going to get a question on step 1 about histone methylation, it better be a question about histone methylation causing muting, okay? And histone acetylation, which removes the histone's positive charge, actually causes relaxation of the coil, right? So acetylation causes histones to not be so positive anymore, so they won't stick to the negative phosphates of the DNA itself, right? The DNA backbone. So then it all relaxes, and that allows transcription to occur because now DNA can be accessed by enzymes. This was actually a pretty big question in, I believe, both UWORLD and NBME about 2 years ago, after which I had added this note into first aid in my original 2019 and the 2020 annotations. So they wrote this here, this topic of thyroid hormone, right? So they wrote now in first aid 2021 that thyroid hormone actually has receptors, right? So thyroid hormone receptors alter thyroid hormone synthesis by acetylation, right? So this is a key thing you need to know. There was a question, I'm almost 100% sure it was an NBME question, but it might have been either AMBOSS or UWORLD as well, where they wanted you to be able to explain how thyroid hormone works, right? And then it was actually related to histone acetylation. That was the answer you had to pick. So dysregulated acetylation, this is another thing that was also talked about in my annotations earlier. Dysregulated acetylation, they've added this here now, is implicated in Huntington disease. So you're going to see that in Huntington disease, one of the pathophysiologies of Huntington disease is that you have dysregulated acetylation, which means you lose the acetylation. Without acetylation, you're not going to be able to activate a certain DNA, right? In Huntington disease, you lose the ability to activate this one thing where you're supposed to transcribe it, and you make sort of a neuroprotective protein. You lose that neuroprotective protein, and that's what actually causes the damage you see in Huntington disease. Very key thing to remember. When you get to Huntington disease, this message will hopefully ring a bell for you. You're going to see that. It was also taught by UWorld, and there have been questions about this pathophysiology of Huntington disease. Very high yield thing. So clearly we can see histone acetylation and methylation are significantly more important on this page than the concept of what is euchromatin or what is heterochromatin and stuff like that. The last thing they have here is histone deacetylation. So histone deacetylation is the removal of acetyl groups, which effectively is pretty equivalent to histone methylation. However, if you get a question comparing histone methylation and histone deacetylation, there's one major difference. Histone methylation, we said that may possibly actually be activating DNA, right? And then it's going to activate transcription of that DNA. But histone deacetylation, no matter what, will always decrease transcription because if, as we said, if you acetylate the histone, DNA opens up and you can do transcription. If you take that acetylation away, DNA becomes very tightened up because acetyl groups were actually lowering the positivity of histone, right? Adding those acetyl groups specifically to lysine was lowering the positivity of histones, so they were no longer attracted to the negativity of the phosphates, right? And then it opened up, so now you could do this transcription. So deacetylation will always lower transcription as opposed to histone methylation, which could possibly activate transcription. All right, so that's the bulk of what's happening in this first page. Really not the most, high yield thing, but this is what happens. Most students, when you read a certain page of first aid, you realize, okay, I got it, I read it, I know it all now, and you don't feel like there was anything too substantial on this page, anything too significant on this page. But turns out there's a lot of significant things on this page that you can actually get a full 20 question test just on this one page alone. Right? There's a lot more to take away than what's actually there, and an image itself tells 1,000 stories. So they have a couple things here, right? They have the picture of your chromatin structure. They have this electron microscope image they're showing you, where you have the heterochromatin, the euchromatin, and then deep inside they're showing you the nucleus. It's full of heterochromatin, right? That only thing you see inside the nucleus, that NU signs for the nucleus, all you see in there, well, the nucleolus actually, we're already inside the nucleus. Keep in mind, we're already inside the nucleus, which is why it's full of all this DNA. The nucleolus, even deeper inside, is all full of heterochromatin, right? Which is completely condensed. Heterochromatin, HC for highly condensed, like it says in the book. That's their mnemonic, so that's something you have to keep in mind. Now, they also mention bar bodies, which are a type of heterochromatin. Bar bodies are that second X chromosome that all women have, right? You're female because you have two X chromosomes. When that one X chromosome is put away, because you don't need to use both of them, the one that's been put away, that's a heterochromatin. That's an inactivated X chromosome, and that plays a big role in genetics when it comes to gender differences, because a woman might have an X-linked recessive disorder that might have some level of penetration just because you're a carrier. But if you muted away the X chromosome that is the bad one, then you won't notice anything. So that's a key thing that happens in genetics. But you can get images like this. You can get an electron microscope image like this, and they can ask you what you would find in the nucleolus, what you would find outside within the nucleus. Right? So all the white stuff is your euchromatin, which is actually available to be transcribed. Right? So eu starts with an E. It can be expressed because you can transcribe it. Right? So that's something else they teach you on this first page, and that's really all there is here. The next page is all stuff for you to do on your own. There's nothing really critical here. Right, this page is showing you nucleotides. It tells you a few things that you need to keep in mind. And as I said, right, one of my key number one advice is always revise things, right? Revise topics all the time. So right now, what topics did we look at? Well, the topic we looked at was chromatin structure, right? Chromatin structure, which had a subset of topics in here, right? It had a subset of topics in here, which were your key words, right? So we talked about the histones, acetylation, methylation, and deacetylation, right? And that there was clearly a difference between deacetylation and methylation. You would revise the fact that we talked about heterochromatin, like the bar bodies, versus euchromatin, right? And when I say revise, I mean like without the book in front of you. Right, this is what I always say. Whenever you study a page, you've got everything, you went through it, you talked about it to yourself. Now, look away from the monitor, and within your mind, without the book in front of you, you should be able to talk about everything we just did, right? Everything me and you just talked about right now in this video, right? The parts of a histone. Such as the lysine, arginine being positive, acetylation removing the positivity, and phosphate being the negative thing. Right? Those are the key things we've talked about so far. Now on the next page we're looking at nucleotides. So nucleotides, this is your second topic, right? So topic number one was chromatin structure. Topic number two that we're looking at right now is your nucleotides. So now with nucleotides, we have a couple things here. First of all, the definition. They have nucleoside, which is a base plus a sugar, and then nucleotide is a base plus a sugar plus the phosphate. And there's more to this definition. In fact, the only thing that is actually called a nucleotide is when you have one phosphate. Okay? As soon as you add more phosphates, those are also called nucleoside. All right? So when you have a DP, right, with the diphosphate, or ATP with the triphosphate, those are all nucleosides, which is why we never refer to ATP as a nucleotide. An ATP, adenosine triphosphate or any other sugar, right, with the triphosphate or diphosphate, those are all nucleosides. And then if there's no phosphates at all, if you just have the base and the sugar, that is also just a nucleoside. Okay, the only nucleotide is when there's one phosphate. So that's a key thing to remember as far as like structure is concerned, right? And this page is clearly all about structure. They then mentioned the deamination reactions that can occur. Now these are going to be pretty critical, as we're going to look later on at the various types of DNA repair that can occur, right? So there's one specific thing that fixes these deamination reactions. You need to undo it. If there's a deamination reaction that occurs in a place where you didn't need the deamination reaction, you have to undo it. So this page is listing out those deamination reactions, which are not so crazy. These changes are actually what normally occur. Cytosine to uracil, adenine to hypoxanthine, guanine to xanthine, and 5-methylcytosine to thymine, right? These changes are not that far-fetched. Now, if you ever actually look at something like this, for example, then you can see where the changes are coming from. Right, so here you have your adenine and guanine. You have cytosine, uracil, and thymine. Remember that uracil is only there because uracil is one of those RNA things. Right, you only find uracil on RNA, and you find thymine on DNA. And the deamination that can occur is basically removing the amine. Right? So when you remove the amine, it becomes something else. So for each of these, if you take cytosine, you actually do a deamination reaction to cytosine. This cytosine will become uracil. So you would remove this right here. Right? That's the deamination you would do to make your cytosine into uracil. Adenine and guanine, these guys are going to become hypoxanthine and xanthine. Right? And we're going to see that later on in your purine salvage pathway. You're going to see this recycling thing that occurs. That recycling involves using hypoxanthine and xanthine, right? Those are going to be pretty key things over there. And then thymine, the only thing you see here with thymine, when you compare thymine and this one over here, these two, right? Thymine and cytosine, one of the key differences between the two is the fact that you have this methyl group, right? So that's the key difference here. You don't have the methyl group here. Right? So when you have 5-methylcytosine and that gets deaminated, it actually becomes thymine. Imagine if this cytosine had a methyl here. That would be the number 5 carbon. Okay, so it's number 1, number 2, number 3, number 4, because there's no carbon here. This is nitrogen. And then number 5. So that's why it's called 5-methylcytosine. If you have a 5-methylcytosine, a cytosine with a methyl like that on the 5th carbon, and you do a deamination, which means you get rid of the amine, then you would get your thymine. That's all they're really showing you there. So this is something that you're supposed to have had, again, like when you were studying this the first time, you were supposed to look up these structures because this page is all about structures. So that's the deamination reaction stuff. They tell you at the very end of this first column that methylation of uracil actually creates thymine. Right? So if you ever get a question where they're showing you that, look at the structures here. Structure of uracil and structure of thymine is exactly the same thing. The only difference is methylation right here. If you methylate that carbon, you would actually get thymine. Right? So that's the only real difference between uracil and thymine. Otherwise, they look pretty much the same exact thing. All right? So that's what they're pointing out for you, which was pretty nice of them. Other than that, they have, of course, the fact that purines are A and G, pyrimidines are CUT, which is as labeled here. So you can always remember pyrimidines as pi, P-Y, or P-I-E would be the correct spelling. Right? And that's because this is a single pi. Right? This is just one single pi. Whereas purines have 2 rings. Right? You have the 2 rings from each of them, adenine and guanine. So that's really all the differences between those two guys, purines and pyrimidines. And then later on they tell you amino acids necessary for purine synthesis. Right? So there's glycine, aspartate, and glutamine. And they have an image showing you where the aspartate is going, where the glycine is going, where the glutamine is going. These guys, though, are extremely high yield. This isn't just as simple as this picture is right here. What you need to know is that glycine is actually giving the CCN that's actually highlighted in this picture over here in your first aid. Right? That entire CCN is coming from glycine. Your aspartate gave that single nitrogen, and your glutamine is going to give 2 nitrogens. All right? So those 2 things are pointed out in that picture. You have to know it in that manner. Okay? You have to know it in that manner because you can get legitimate full-blown questions just about that one stupid little topic. And that's how step 1 actually is, right? That's what step 1 does. Moving on to the next page. The next page is about your next topic, which is your de novo pyrimidine synthesis, right? So this is another topic that you're going to be looking at right now, which again, pyrimidine and purine synthesis, which again is something that you need to be able to explain. So if someone came up to you, another med student, one of your study buddies came up to you and they said, hey, explain to me how you do de novo synthesis of pyrimidines and purines, you need to be able to literally within, without looking at the book, you need to be able to explain that. You need to be able to talk about it without hesitation, no issue whatsoever. So if you look at the diagram they've given you, they've complicated, in my opinion, they've complicated this diagram for no reason. By mentioning all these medications right now, which you are going to see eventually, you're going to see it as the course moves on, you're going to see it as the book goes on, but it's not something you need to know right now. It's not the point of this chapter, right? This is molecular. Why are we looking at medications? Well, of course, yes, these medications interfere with things here, but that's what you're going to learn when you look at those medications and the case scenarios where you would need to use those medications. For now, you want to focus on the diagram without the medications because you're learning the diagram, you're learning the pathway, right? So it all starts off at the very top with ribose 5-phosphate, as you can see in the center, right? That ribose 5-phosphate gives you PRPP thanks to PRPP synthetase. Super easy so far, right? That was very, very easy. There was nothing complicated about that. R5P, which is ribose 5-phosphate, gave you PRPP, and that's an easy thing to remember the name of the enzyme that's involved over here. That enzyme is PRPP synthetase. It synthesized PRPP. Super easy. Now, there's a couple details here that are missed, all right? PRPP synthetase gave you PRPP, but there's more that's going to happen. Now, there's another enzyme, enzyme number 2, for the second arrow, is going to actually create something called 5-phosphoribosylamine. Okay, this 5-phosphoribosylamine is what's actually going to take the things that were being done on your last page, the nitrogens that are coming from glycine, aspartate, and glutamine, and the carbon that was coming from your THF, as you saw in the same diagram. So in your previous page of first aid, where we were looking at the nucleotides topic, there was N10-formyl tetrahydrofolate on the bottom left, giving you the carbon, all of those guys, right? So these are the things that are being donated into 5-phosphoribosylamine. So I'm going to write over here, plus the nitrogen and carbon sources. And what does this give you? This is immediately going to yield inosine monophosphate, IMP, which you can see in your diagram, right, on the right half. Right, that's the part that's giving you purines. You have your IMP. So you see how there's a bunch of arrows that they're not filling in with details? Those details you do need to know. Right? It's not like it was completely irrelevant detailing. No, it was pretty important detailing. In fact, at one of those points, even 6-mercaptopurine or azathioprine, which are pretty much the same medication, works. Right? Something happens there. And then after IMP is made, you could take something mycophenolate or ribavirin to stop production of GMP. Again, you don't want to complicate this page right now if you're new here with the medications. You're going to learn the pharmacology later on, and learning pharmacology is a whole different topic, right? That I'm going to make a video about it later on, talking about how to master pharmacology, because it's definitely one of the most top difficult things for med students. Right now, what you want to understand is this is what's happening to make IMP, which is on the right half of this de novo synthesis diagram. Now, one of the key things here is, of course, your IMP will eventually yield your GMP and AMP. Right, like that. That's all you're going to get from IMP. Whether you added ATP to give the AMP aspect or added GTP to create the GMP aspect. Whatever you're using, that's what's going to happen. Now, there's something critical here. One of the key things you need to remember is that all of this, as it's accumulating, can actually come back and inhibit the second enzyme. Enzyme number 2, which I told you was called PRPP aminotransferase. Right? Obviously, it's not PRPP synthetase because PRP synthetase was enzyme number 1 over here. Right? Enzyme number 2 is your PRPP aminotransferase. This, as you could tell, is a negative feedback. It's going to stop it. Right? So if this is adding up, if this is accumulating for whatever reason, one reason could be that you aren't doing what you're supposed to do with these amino acids. You're not making protein, right? You're not making DNA, etc., etc. So if this is all accumulating for whatever reason, you would stop it. You would stop making more. All right? So that makes PRPP aminotransferase a pretty high yield important enzyme. The number one place where this actually happens is Lesch-Nyhan syndrome. This is going to be significant. It's on the next page where they show you in Lesch-Nyhan syndrome you have defective purine salvage due to the absence of an enzyme HGPRT, which actually converts hypoxanthine to IMP and guanine to GMP. Right, so you see the IMP and GMP here. When you have this deficiency, when you have a deficiency of that's seen in Lesch-Nyhan syndrome, you have excess purine synthesis because you're going to have more PRPP aminotransferase activity. In first aid 2021, they call it PRPP amino transferase. PRPP amido transferase is the same thing. Don't worry about that aspect of the naming. But this is what happens in Lesch-Nyhan syndrome. This was actually a question a couple years ago in UWORLD, where they were like, what enzyme would have increased activity? And of course, everyone chose PRPP synthetase, but that didn't make any sense. But I get why they chose it, because that was the only enzyme people had heard of. Now you see it in first aid 2021, about 4 to 5 years later, that it is the enzyme that is responsible for excess purine formation, which actually ends up causing the gout and everything, and hyperuricemia and whatnot that you see in Lesch-Nyhan syndrome. Right? So once again, because you have defective purine salvage due to absence HGPRT, you're unable to convert hypoxanthine to IMP and unable to convert guanine to GMP. So what happens in these people is that you will not have GMP and you will not have IMP. Why? Because you don't have your purine salvage, right? So it's, remember, HGPRT is supposed to convert hypoxanthine to IMP and guanine to GMP. That's what it's supposed to do, and you can see that in the image here. It's not going to be able to do it. Right? It's not going to be able to do it, so you don't have these guys, so you do not have this negative feedback that's stopping PRPP aminotransferase from working, so that means it keeps working. So you're going to try to keep making this, and this is going to end up being a really bad cycle for these patients, right? So they end up having excess uric acid production due to this excessive purine synthesis, and then they're going to see all the symptoms that they're going to present with. Now, in questions about Lesch-Nyhan syndrome, so let me write that now since we're talking about Lesch-Nyhan syndrome. All right, so actually let me continue that down here. All right, so Lesch-Nyhan syndrome, another topic we're looking at today. The symptoms that they've written now as their HGPRT mnemonic, right? Hyperuricemia. Well, obviously, because you're making too much amino acids, right? You lost this negative feedback. You no longer have this negative feedback. It's not going to work. So you're going to make too much amino acids, so that's why you get hyperuricemia. The hyperuricemia leads to the G, which is gout. One of the symptoms that you're going to see in these patients is that they have this aggression and self-mutilation. So the P is for pissed off, right? Pissed. These are going to be young kids. Keep in mind that Lesch-Nyhan kids are showing up like really, really young, like age 2, 3. They're having these issues that are very abnormal. You can even start seeing symptoms while they're under age 1 year, because the R stands for the red-orange crystals that you might see in their urine. Now that's usually a giveaway. So STEP may not even mention it, because if they mention that they see these things in hyperuricemia and the red-orange crystals in a maybe 9-month-old diaper, right, 9-month-old kid's diaper, then you're simply going to go ahead and pick Lesch-Nyhan syndrome. Or they could be assholes to you and they could tell you there's a red-orange crystal, and then they'll ask you what's in this red-orange crystal. So the book does tell you that it is mainly composed of sodium urate crystals. So you have to pick that as an answer choice, right? It's composed of sodium urate crystals. And then the last thing, the T stands for tense muscles, which is dystonia. Right, so tense muscles, you're going to see that as a symptom in the child as well. They could also have macrocytosis, intellectual disability, and so on. One of the most fascinating things about the P, the pissed off aspect, is their point of self-mutilation. Right, they can do that. They can self-mutilate, and that's something that you never see in a child. Right, so when you see that, that's also essentially a giveaway for HGPRT, because no child self-mutilates. It's just not a thing. Treatment for HGPRT is mainly going to be to use allopurinol or febuxostat, and that's really all there is to this topic. There's one other topic here, adenosine deaminase. All right, so adenosine deaminase deficiency. So before we talked about adenosine deaminase deficiency, right, we need to understand the general aspect of your purine salvage deficiencies, purine salvage in itself. But before I move all the way down to that, let me go back up to what we were doing before, which was your de novo synthesis, right? De novo pyrimidine and purine synthesis. So I showed you the ribose 5-phosphate yielding ultimately IMP, GMP, and AMP to make your purine synthesis. All right, now let's look at the pyrimidine side. So on the pyrimidine side, what you're seeing is the glutamine, which was one of your donators of nitrogen from earlier, right? Glutamine is going to combine with CO2 and use some ATP through the help of this enzyme CPS2, carbamyl phosphate synthetase 2, which by the name carbamyl phosphate synthetase, you can tell it's going to make carbamyl phosphate. Right? So that's the first step. This is a very high yield step. You have to remember that this is enzyme CPS2, carbamyl phosphate synthetase 2. There is a CPS1 found in the mitochondria that you're going to see in the urea cycle. That's going to be later on. The urea cycle is very intimately and closely related to purine and pyrimidine synthesis, because even the ribose 5-phosphate we see here, right, that's something that's related to another cycle, which is the HMP shunt. In the HMP shunt, you actually make ribose 5-phosphate. That's where this is coming from. So again, when you're studying biochem, any biochem, especially biochem metabolism, there's a lot of other sections of the book that are relevant, and you have to make sure you do those jumps, and I'm going to make sure I point some of these out to you. So those are the jumps I'm pointing out to you right now. If this is your first time studying for step 1, you don't need to go to the jump. But if you're already done studying, or you've done some studying and now you're revising, you should take the jump, go check the urea cycle, go check HMP shunt, and then come back here. So like you would pause the video, do the jump, check what I'm talking about, come back here, continue. So here's CPS2. CPS2 is actually in the cytosol. They have a mnemonic for you on the bottom right where they write cytosol, the number 2, to remind you CPS2. Right? CPS1 is in the mitochondria. This carbamyl phosphate then combines with aspartate to give you orotic acid. Orotic acid is another huge topic that you're going to see later on in hematology as a topic of orotic acid urea. Right? In orotic acid urea, you have a deficiency in the activity of the next enzyme, UMP synthase. What UMP synthase actually does is that it makes UMP. Again, by the name you can tell. So these enzymes are named perfectly. UMP synthase makes UMP using orotic acid. So in patients with orotic acid urea, their urine is full of orotic acid because they're unable to make UMP. There's something very critical here that happens to them. Think about what would happen. Well, if you're looking at the diagram, you can see that the orotic acid is supposed to give you UMP, and that UMP will eventually give TMP and CTP, right? So you're basically gonna make thymine and cytosine, which are gonna be important for DNA replication. So in those patients with orotic acid urea, they're gonna have anemia because they can't do DNA replication. So they're gonna have that anemia that's very critical for you to use to differentiate orotic acid urea from another disorder we're going to see that also involves orotate. Right? So a very key thing to keep in mind. Orotic acid urea is a deficiency of UMP synthase that you're going to see in hematology. Very easily treated. All you have to do is give them UMP since they can't make it. Right? So very straightforward. Now, once you make UMP, you can then convert that to UDP, and now you need a very critical enzyme, ribonucleotide reductase. See, UDP is missing the D that you see later on. See, now after ribonucleotide reductase, there's this thing, it's called DUDP, which is deoxy-UDP, and deoxy things are for DNA. UDP itself was not something you could use for DNA synthesis, right? So if you don't have ribonucleotide reductase activity, you can't make DNA, which is why they're pointing out a medication here that I do want you to keep in mind, which is hydroxyurea. It'll prevent you from using ribonucleotide reductase activity, and you will not be able to do any DNA replication because you can't make your deoxy-UDP. All right. This deoxy-UDP then becomes deoxy-UMP, and then another key enzyme, thymidylate synthase, makes thymine, right? That's why it's named thymidylate synthase, to give you DTMP, and you have another key medication here, 5-fluorouracil, as well as capecitabine. Right? Both of these very, very important. The last enzyme here that's super important is dihydrofolate reductase, which has its own set of 3 different medications used in 3 different species: humans, bacteria, protozoa. Right? 3 different, completely different organisms. So for humans, we're going to be concerned with the methotrexate. Right? So you use methotrexate to prevent dihydrofolate reductase from doing its job. You can no longer make THF, and then you cannot make DTMP, which will also inhibit DNA synthesis. all of which would be things you would need to do in people who might have some kind of cancer where they're making too much DNA. Right? One of the key mechanisms we've used for the longest time to treat cancer is to just stop DNA replication. If you could stop DNA replication, the cancerous cell cannot infinitely replicate itself. In today's time, DNA replication is still one of the oldest things we use, but in today's time we have new ways of stopping cancer cells from developing. Right? Part of the things that I see as a clinical research coordinator, the job I do here in New York, there's a bunch of new medications that are coming out that are also in first aid now. You're going to see them in the pathology chapter under oncology, in the pathology chapter under neoplasia, where they mention things that are targeting random receptors that only cancer cells need, like the PD-L1 ligand, right, and the PD receptor itself. So those are some things that you're going to see later on when you get to neoplasia under pathology. But that's really all there is to pyrimidine and purine synthesis, and if you do this correctly, you can make DNA. If you stop any of this, you cannot make DNA. That's really all there is to it. So that's why this is such a high-yield thing, because it's related to cancer, and you can also use this to kill, as you see, bacteria and protozoa, because if you kill their ability to replicate DNA, they'll die. Right? So that's where the trimethoprim and pyrimethamine are coming in. Now, trimethoprim, you have to keep in mind, trimethoprim, which is usually taken as TMP-SMX, trimethoprim sulfamethoxazole, becomes a sulfa allergy medication, so you have to be very careful in patients who might be sulfa allergic. You actually mess up their ability to make DNA as well, right? But mostly in the bacteria, not the human itself, but you kill their normal good bacteria. You know, there are bacteria you have that you need, especially in your gut, so that's going to be a pretty big deal for those patients. All right. So again, now the last thing, purine salvage deficiencies. So in your purine salvage, right here again, you can see the same at the very top. You have ribose 5-phosphate, which uses de novo synthesis to make IMP. IMP can make AMP and GMP. So we're only looking at purine salvage, not the pyrimidine salvage. Right. So this is why I pointed out to you the entire purine production. So that's what we see at the top. We see PRPP gave you IMP, GMP, and AMP. That's your top part right there. Now, they can become nucleosides if you take the phosphate away. You need one phosphate to be a nucleotide. As soon as you take it away, you become guanosine, inosine, or adenosine. Those are your nucleosides. To recycle this, the guanosine would give you a free base called guanine, right? And your inosine and adenosine both give you a free base called hypoxanthine. PRPP, right here, would combine with either hypoxanthine or guanine using the enzyme HGPRT to recycle and remake GMP or IMP, as you can see right there in the diagram on the left half. If you have Lesch-Nyhan syndrome, you don't have HGPRT function. So what do? Well, now your guanine and hypoxanthine will become xanthine. The xanthine will then go to become uric acid, which can lead to uric stones. Right? And those are the things you see, right? Hyperuricemia and gout, those are the things you're going to see in patients with HGPRT deficiency in Lesch-Nyhan syndrome. Right? Xanthine oxidase is the main enzyme right there that's eventually creating uric acid, and then you have urate oxidase, also known as respiratory as a medication as well, that can convert uric acid to allantoin for excretion in the urine. Right? It becomes more water soluble at that point. But if you have too much of this going on, then you don't have enough time to actually make all of it into allantoin and actually nicely pee it out. Right? And in HGPRT deficiency, you're going to have this happening too much. So that's the only real purine salvage deficiency issue as far as hyperuricemia is concerned. The other one is ADA deficiency, right? Adenosine deaminase deficiency. In the book, they tell you that adenosine deaminase is required for degradation of adenosine and deoxyadenosine, which is just the one that has the DNA aspect of it. If you don't have ADA activity, you end up having too much deoxy ATP. Right? That's what they write down for you. But you don't see that in the image per se. You don't see it in the diagram. Right? But a lack of ADA will lead to too much DATP. Now, this DATP actually ends up being toxic for lymphocytes, right? Because now with all the excess DATP, you actually end up having a decreased activity of ribonucleotide reductase, which we just said in the previous page that it was one of the key enzymes for making DNA. right? The last three enzymes under the left half of pyrimidine synthesis, de novo pyrimidine synthesis, are ribonucleotide reductase, diamidylate synthase, and dihydrofolate reductase. These three enzymes are very critical for making pyrimidines, right? You need your pyrimidines and purines to make DNA. Here, in these people, with the excess DATP, it's almost like there's a negative feedback that's going to occur because of the excess DATP, that's going to lead to a reduced activity of ribonucleotide reductase. Now you have a decrease in DNA precursor cells, and the first set of things that get affected by the reduction in your ability to make DNA is going to be your lymphocytes, because you need to make lymphocytes. So this leads to one of the major causes of autosomal recessive SCID, severe combined immunodeficiency. Severe combined immunodeficiency can also be X-linked, and you're going to see the rest of that when you get to immunology. Right? And in immunology, I'll break down this once again. So there's plenty of chances to revise these things, right? There's plenty of chances to revise these things. So every time these things come up, when you get to immunology and you see the same ADA deficiency and severe combined immunodeficiency as one of your immunodeficiency syndromes, you should come back here and look at this page again and make sure you understand the diagram, the chart. You should know where things are going, and don't be confused about any of this. At some point, every diagram should be something you can reproduce. This is also a diagram. This entire purine salvage deficiency diagram is something that you should be able to sit down and reproduce without the book in front of you, right? So this is something this is a point you reach eventually when you're doing UWORLD questions, when you're doing whatever. Every time you're studying, you come back to this page, you create a better mental image of this page, and it sticks to you, all right? So we'll stop here for now, and then later on, after the next video, we'll go on to your genetic code features, which is going to be very critical in the discussion of genetics later on in biochemistry.